This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 421, recorded on December 23rd, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,500 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code MICROBE. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Stay tuned to hear how you can win a brand new copy of Principles of Virology 4th edition. Joining me today right here in New York City, Dickson Despommier.
Hello there, Vincent Racaniello.
Well, what's the formal greeting for it, Dickson? Any particular reason? Are you mad at me?
Not at all.
You know, when your parents call you by the full name, it's usually because they're mad at you, right?
For all the good things that you've done for me over these many years, I could never be mad.
Dixon, it's warmed up a bit here.
Yeah, you know, it's unseasonably warm.
Well, 8 degrees Celsius. I don't know if that's unseasonable.
It's unseasonable for this time of the year, I think.
It's a bit cloudy out here. You know, but previously it was sub-zero.
We had a wave of very cold weather, and then all of a sudden, the next day it went up to 50 degrees.
It's good to have a little warmth here. And I'm not talking about you. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it's also pretty pleasant here. I don't think it's that unseasonably warm, but it's 45 Fahrenheit, 7C. But beautiful, clear blue sky.
Really?
At least it was last time I looked. Yeah.
Yeah, we don't have that here.
No clear.
It's supposed to get really nasty tonight into tomorrow with wintry mix
Really? Wintry mix.
Yes, that's definitely seasonal.
Wintry mix. Huh. Wow. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. It's 32 degrees here, and everybody should know what that means. That's in Fahrenheit, 0 degrees Celsius.
Yeah, it's cold. Now, I talked to Michelle Swanson yesterday. She said you had some snow on the ground, right? Or is it gone? Is it gone?
No, it's still all here because we got so much.
It never goes away out there.
Streets, yeah, no, it does. The sidewalks and streets are mostly clear except for the places where there were parked cars and the plows had to go around them, or where the plows have made piles and things like that.
How many inches did you get originally?
Oh, I want to say 7 or so.
Wow.
It's more than a dusting.
Yeah, we still have snow on the ground up here.
Well, we don't.
Not too much here. Well, you know, it's the holidays, I guess, all over the world. But here in New York City, it's a destination, and a lot of people come. And one place that many people want to visit is the TWiV studio. And we've had a couple of visitors. Last week, I had some German students visit. This morning, Dickson had some South Korean students. And today, for TWiV, we have a faithful TWiV listener who's written in on multiple times. He's from the law firm of Olive Law Group in Cary, North Carolina. Nathan Letts, welcome to TWiV.
Great to be here. Thank you.
My pleasure. Nathan is no stranger to these parts. Now, you grew up in northern New Jersey, right?
Yep. Grew up in Glen Rock, New Jersey, in Bergen County.
Which Dickson knows.
Very well. I have a good friend who lives just next door to you.
Where'd you go to college?
Went to Hamilton College in upstate New York.
Oh, great school.
It is a wonderful school.
So there's some college in Hamilton, New York, and then there's Hamilton College, which is in where? Is it
so Colgate is in Hamilton,
New York.
Another small liberal arts.
And Hamilton is where?
In Clinton.
Clinton. Right. Then there's also Clinton College, probably. We won't go there.
In Hamilton County.
You got a PhD in chemistry, right?
So I was a chemistry major there, and then I taught at a boys' boarding school, taught chemistry and physics and geometry, and then went to graduate school at Columbia in chemistry.
Wow, chemistry. That's
cool.
Great. And roughly what years is this?
So I started in 1986 and graduated in 91.
So after you got your PhD, did you go right to law school?
In effect, that's what I did, and so I basically was working at a law firm, Cooper and Dunham, that actually does patent work for Columbia.
Yeah. Yep.
And John White was my mentor.
Oh my gosh, he wrote our transgenic mouse patent.
Yeah.
So
he most famously wrote the Richard Axel co-transfection patent that was bringing in huge royalties to Columbia.
Huge. I know, because Saul Silverstein, who is next door to us, not here today, but he was one of the big inventors on that, and he has many fancy cars. As a consequence of that.
And a fancy house.
Yeah, John. I was telling my Steve Goff, the neighbor, that John White and I worked on that and a couple of other patents a lot. It was a lot of fun, and I think the law firm is down at Rockefeller Center, right?
Yep.
Went down there a couple of times. Those were in the days when we had to hand deliver paper.
Right. No, and we'd also, not only would you hand deliver paper, but we also had to file by carrying things to the post office at 11:30 at night. So we'd take the subway or take a cab down to 34th Street and drop it off there. Now we can do that all electronically, which is a great thing.
And where did you go to law school?
I went to Fordham Law School at night.
Mm-hmm.
So
while you were working at Cooper and Dunham?
Yeah, so I lived midtown and sort of would walk a triangle from my apartment on 46th Street to Rockefeller Center and then up to Lincoln Center and then start the whole thing over again the next day.
And now you're in North Carolina, not too far. You live in Chapel Hill, right?
Yep.
And I saw you there a few months ago when I visited. And you do intellectual property, dealing with, what, chemistry mainly or biology?
It's a blend. I mean, one of the things when I was in graduate school...
Or an emulsion?
Yeah, maybe an emulsion is better. So when I was in graduate school, I had a cousin who's like, I only know two scientists, you and a guy by the name of Alan Maxim. Do you know Alan Maxim?
Yeah.
And I was like, Maxim Gilbert, sequencing fame. So she's like, so she set up a meeting with me to meet with him my third year in graduate school. And I studied like back and forth all of the, DNA sequencing reactions and things like that when I was there. And he was like, I don't do that anymore. I do really molecular biology. And he was talking about sort of how exciting it was in eukaryotic transcription around 1987. And so that kind of really made me want to get into biotechnology and life sciences. So
how long have you been listening to TWiV?
So I started, I think it was the summer of 2010.
Wow. And are you a podcast listener in general, or?
I've become more so.
Mm-hmm.
So I listen to the TWiVo and TWiM, and then I've got some sort of kind of political or like Signal is a good one for if you want business and biotech is a good one that I listen to from Stat News and.
Great.
Cool.
All right. Well, don't be shy. Jump in whenever you want. You got a mic. You got headsets.
Lawyers have never been known for their shyness.
Yeah, it's true. Kathy,
Chemists, on the other hand.
Kathy, you want to tell us about ASV?
Sure. A reminder again that this is a really good time to be working on your membership for ASV and why you want to be a member, besides all the particular benefits of just being a member. If you want to attend the meeting and you're a student or postdoc and you want to apply for travel funds, you need to be a member. And if you're going to sponsor anyone's abstract, and all the abstracts need to be sponsored by a full paid-up member, then this would be a really good time to either renew or join ASV for the first time. And the deadline for those travel awards is February 1st, which is the same as the abstract deadline. So that's why I'm telling you now, because we know that you're going to wait to do your abstract to the very last minute, but you can do this membership easily now. So I've already written 2 sponsor letters, and I'm about to write a third one. And so you can start asking people to help you get that membership. And speaking of abstracts, it's not too soon to start thinking about it. And I want to remind you that on the website, which is reachable from asv.org, click on annual meeting and then click on annual meeting again and then bookmark it. There's a list of topics that you can submit your abstract. You get to choose a first and second choice for the workshop that you think would be appropriate. And we've added a few topics and modified a few topics for this year. So you want to take a look at that and be thinking about that now, even before you're thinking about the real detailed content of your abstract. You can also register for the meeting now. You can think about the dorms or the housing if you want to stay in a hotel. And one other thing is that there's the career planning workshop for junior virologists, which is free, but it's limited to the first 100 students and postdocs that register. So we're expecting that might fill up, and you might want to look into doing that now.
All right.
Cool.
I also want to let you know about a joint ASM-ASV conference on the interplay of viral and bacterial pathogens. It's organized by Christian Vobis, Stephanie Karst, Julie Pfeiffer, Stacy Schultz-Cherry, and Vincent Young. And all of them have been on either TWiV or TWiM. I think it's cool. That's our goal. One day, any virologist or microbiologist mentioned will have been on one of these podcasts. It's a lofty goal, right? This is going to take place from May 1st to 4th in Bethesda, Maryland. It's one of the hottest new topics in biomedical research, trans-kingdom interactions between viruses and bacteria in the gut and elsewhere, of course, and how they influence health and disease. As I said, it's co-sponsored by ASM, the American Society for Microbiology, and ASV. It's a brand new conference, and it's going to bring together scientists from different disciplines to talk about cutting-edge research related to this hot topic. Some of the sessions will include understanding the cellular and organismal intestinal host response, the interplay between enteric viruses, bacteria, pathogenic bacteria, and the microbiota, novel approaches to model intestinal infections, and much more. And if you listen to the last TWiM, you'll hear all about this sort of thing, interactions between viruses and bacteria. Jennifer Baumberger from UPitt. There is a deadline for the abstract submission. It's February 21st. You have a little bit of time. Sounds like a cool meeting, though. It's in an easily accessible place, Bethesda, Maryland. And I'll post a link to the sign-up sheet in the show notes. So check that out.
Right on the red line. Is it now? Is this the? There was an ASM-ASV joint conference on Zika, wasn't there? And
there was.
Was that
Last
the
June.
first? Was that the first time they'd ever done a joint conference?
It was.
So it's nice that they're continuing this.
Yes, it is good because, that was organized by Lynn Enquist and Grant McFadden. So it's nice that it's not going to be a one-off thing, and they want to continue it.
That's great.
Part of the détente between the two societies.
Excellent.
Détente.
You know what that means, Dickson?
I do, but I think it's a lot more than that.
Oh, no. You don't know the history, Dickson. We won't go into it now, but there's a long and sordid history.
Well, you remember
it was. Let's just say it was a lytic separation.
Well, I am a member of the American Society for Parasitologists, so they don't even speak to us. So,
what do you mean us?
We could
us
never
meaning,
have a conversation.
Us meaning who? Us?
Is it the
Us meaning-
virologists
ASM
or the virus?
ASM. ASM was the group that would never even talk to us. So,
all right, we have some follow-up. Busy week for follow-up, despite people traveling. Jen is from Mount Sinai. She's a graduate student there. She does our timestamps. She listens every Sunday and emails me by Sunday evening the timing for every show note and so forth, and I put
which
it
is
in.
Awesome.
It's really great, and of course we're going to lose her when she graduates, which will be probably next year.
Mm-hmm.
But anyway, in the last show notes, she wrote, "My favorite Kathy quote ever is at 46:55, 46 minutes and 55 seconds." So I figured Kathy would look that up.
Yes, because I thought maybe it was from TWiV 420, but that didn't make sense. So I went back to TWiV 419, and it was when I said, "Once again, I'm thinking," and pedant, saying that. And it was the second time in the podcast that the dialogue had gone pedantic. And so I confirmed that with Jenny, and she said, yes, that was my thought exactly at that point also.
Listen, it's important to have the right terms.
It is.
Jenny, you got to know. And as I often say,
terms like pedant,
because we write we write a textbook, you have to use the right terms throughout. So,
that's right.
And I will not hesitate to give you my opinion about the use of certain terms. It's my job here, right? It's our job, actually.
It is your job.
Same with me when I edit people's things. I'm pedantic about when they use as and they really should be using because or since.
It's not necessarily a bad thing, right?
Right.
All right. Connor writes. Connor is friend. He's a postdoc over in Glasgow who's been on the show. Some follow-up to a recent discussion, further to your discussion on the last TWiV about the ongoing outbreak of mumps in the U.S.
Say monks.
I want to stress that the mumps vaccine, Geraldine strain, used across the U.S. and most of the world, is one of the best vaccines that we have and is incredibly safe with over 50 years of experience. Over this time, it has led to a 97% reduction in cases of mumps and has near eliminated associated hospitalizations for complications such as meningitis, encephalitis, and has and even the very painful orchitis.
Right.
Look it up if you don't know what it is. However, outbreaks continue to occur, and there have been particularly large ones, 2005, 6, 2009, 10, and now 2016. The reason why we are experiencing outbreaks of mumps now, which includes those immunized with 2 doses of the MMR vaccine, is likely a consequence of multiple factors, as you suggested. These include continued presence of unvaccinated individuals, waning immunity, very close contact of individuals, and minor antigenic differences between vaccine and circulating viruses. Although mumps virus is serologically monotypic, the vaccine strain genotype A is considered extinct, which taken together could lead to a perfect storm for mumps. The issue of waning immunity or secondary vaccine failure is complicated. Perhaps the vaccine is not as protective as it could be, or there is little boosting of natural immunity as the wild-type virus circulation wanes. While the effectiveness of a 3rd dose is not clear, he gives a link for that. One strategy would be to generate a completely new vaccine against the virus that provides stronger, more durable protection against disease while remaining safe. There is a precedent for this because infection with wild-type mumps virus provides near lifelong immunity against mumps. Of course, this comes with a cost in terms of disease, and getting the balance right is essential. There is so little understood about mumps virus pathogenesis and immunity that designing a vaccine rationally that is both efficacious and safe would be very challenging. For example, we do not know the viral and host molecular determinants of disease. We don't know correlates of immunity, how important are antibodies compared to T cells, for example, and we don't know very much about the fundamental biology of the virus in terms of tropism, pathogenesis, and transmission. This is in part due to a lack of model systems on the virus and host side. I aimed to address part of this disparity as part of my PhD project in the Dupre lab and established a reverse genetic system straight from unpassaged material from a mouth swab from an individual in the 2009-2010 New York outbreak. Modified recombinant viruses from this system and others like it will prove invaluable to allow us to investigate the critical questions discussed above, which will be important in the development of new vaccines. And he sends a link to a review on mumps pathogenesis and molecular biology that he published recently. Thanks for the great show. Cheers. Connor in grey, rainy, but always beautiful Glasgow, Scotland. Yes, sir. Alan, can you take Chris's?
Sure. Chris writes the article by C et al. Entitled Generation of Influenza A Viruses as Live but Replication Incompetent Virus Vaccines is very interesting. Although this is the one we just talked about last episode, it is not so different from the generation of infectious replication defective viruses used in more conventional gene transfer gene therapy studies. The transgenic cells containing cassettes for expression of orthogonal tRNA, tRNA synthase, and an amber codon containing GFP are not so different from the packaging cell lines used in the generation of retroviral, adenoviral, or AAV-based vectors, where replication defective vectors are desired for safety reasons. Are replication defective vaccine viruses likely to be more effective at stimulating effective immune responses than a fully replication competent one? Only time will tell, but I'd be surprised if they do, as in most cases the desire is to mimic the effects of natural infection, albeit with a less virulent strain. At least vaccines based on the orthogonal approach to infect cells, meaning they possess a distinct advantage over inactivated vaccines. Perhaps they may prove to be particularly useful where an immune response different from the one stimulated by natural infection is required, where natural infection fails to simulate an immune response that's capable of clearing the host of the pathogen or protecting the host from reinfection, HIV, for example. Wishing you all a very happy Christmas and healthy and successful 2017. And Chris is at Middlesex University.
Happy Christmas. Isn't that what they said to Harry
happy
Potter?
Happy Christmas.
Happy Christmas, Harry.
Right.
Yeah, I mean, I think it is not so different, but it's new technology. That's why I was, and maybe we were excited about it. But yeah, it's sort of like a replication defective virus that you produce in a packaging cell line and so forth.
Right.
And we'll see if they work. But stay tuned, Chris. We're continuing this arc today.
Yes, we are.
As yet another approach to a similar kind of vaccine.
Mm-hmm.
Now, Nathan, I was just thinking, in all this technology, we should have you back sometime to discuss the CRISPR patent issues, right?
Absolutely.
Would you like to come on and talk about it?
Sure, I could do that.
You could do it by Skype from North Carolina, right? Do they let you Skype out of the law firm?
We could do that, or I could do it from home.
Yeah. Now, that's an interesting issue that we shouldn't talk about because we don't know anything about the legal implications anyway, so that would be fun. Kathy, can you take Steve's, please?
Sure. Steve writes, "Hi, Vincent. I've just been listening to the latest TWiV and was pleased to hear that you are interested in my old sparrow story and like the New York City street trees pick. A great way for me to explore the city from here even. I like popping into the Smithsonian Natural History Museum tour also. Isn't the web marvelous? I just heard you saying about the advertisements coming to an end after Christmas and wondered if you had thought of the ads that are still in the earlier podcasts. I don't know if having the ads permanently available was part of your deal with advertisers, but it strikes me that having them playing in the archives all the time is not going to encourage advertisers to give you fresh business for your funds.
Wow.
You might like to look into editing the archive programs before the backlog of adverts gets great enough to make this a considerable chore. All the best and Merry Christmas to you and the Twix teams, hoping for 2017 to be a much better year for all. Perhaps the new movement to bring some fact-checking and real truth to news on the web will start to turn things around and herald a new era of ethical behavior. Perhaps." And a smiley face. Steve, "With a crisp blue, fine winter's day just getting underway outside my window." So what do you think about that, your old ads?
So you're going to subtract the ads?
No, because the advertisers actually don't care anything that's more than a month old.
Right.
In fact, they're mostly interested in the weeks after the 1 week after release of an episode, and they want to know how many downloads you have. That's what they focus on.
Right.
There's no way to really get the ads out of the old advertisements without a lot of work. But that's actually one of the benefits of advertising on a podcast. And when people go back, they hear the ad, and it's got a long tail, right? So that's what we all do, and advertisers do not advertise because of that. They always want fresh ears, and because there are always new people listening and so forth.
Right, and this is not a new problem. If you look up old copies of the New York Times, before the internet, you'd go to the library and look this up on microfilm or even in the paper version, and there are the ads.
I love looking at old ads
from centuries past or decades past.
It's fun.
They're a lot of fun, and they're really also an important part of the historical record.
Yep.
In that, I don't know that our Drobo ads or what have you are going to be an important part of the historical record in the future, but one never knows.
But, and Steve, yes, Dickson, were you going to say?
Oh, no, Nathan. But Alan, I mean, for things like television and movies, I mean, that there's a whole business of reselling the archive, old, sort of catalogs of things, and then they layer on top of that current advertising.
Yeah, sure.
But of course.
Yeah, we don't do that.
Well, should we get to a point where we're, making a business out of old TWiV episodes, then it might be worth going into those episodes and replacing those ads with clips of new ads.
Yeah.
That would be a good way to do it.
And I wanted to say, Steve, with respect to your trees, New York City trees map, which is just awesome. So right now, the TWiV studio is at 168th Street and Fort Washington Avenue. You can go and look at all the trees that I see every day, and Dickson as well. And it's really cool. You can have a street view of the front of the building, and you can see all the trees around as well. You can get a little bit more involved in where we are.
Some of the most interesting sitting in the porcelain throne room was to see all of the wallpaper made from old Sears and Roebuck's catalogs.
Yeah.
And you see all the ads for weight loss, and you see a capsule containing a proglotid from a tapeworm, for instance, and stuff like this. So, yeah, it's absolutely fascinating stuff.
Hey, Dickson, we have one more from Steve.
We do. Would you like me to read it?
What do you think? I'm talking to you just
No, just a point of information.
Yes, please read it.
Okay, so Steve writes, "Hi again, Vincent et al." So he acknowledges the rest of us in this one. "I expect you will be deluged by geology and physics buffs after your after your changing Earth's rotation comments on this week's TWiV, but I had to do a double take when I heard it being said that the human race had gained a whole 6 hours of extra day in mere historic times. Conventional geology has long had it from correlating, no pun intended, radioactive decay with the stratigraphical record and the daily growth rings on fossil solitary rugose corals that the planet has only gained about an hour in a day length since the Carboniferous period some 350 million years ago. As you can imagine, this is well-trodden ground in the debate between scientists and creationists, so it is unlikely that the new story that you referred to can be correct. The subject might make an interesting episode, for TWiVo. Your podcasts are a very good memory aid for us old guys. Cheers. Steve.
Day now even brighter, wintry blue than my last note.
That's right.
I think they were very close together. All right. Thank you all. Yes.
So was that a misinformation?
Oh, I don't think so. I mean, that was a scientific paper that the calculation was based on, right?
Well.
And that was Rich's pick last week, I believe.
At the rate that's going, though, however, by the time I get to be old, and that hasn't happened yet, the Earth might even stand still. And I've seen that movie. It's not a pleasant sight.
No.
Well, I am not someone to comment on this. This is not
right.
my
So, in other
field.
words, this finding is probably not quite right.
You think?
Right. Maybe they misplaced a decimal point or something.
Maybe.
An hour instead of 6. Well, it's pretty close. It's less than an order of magnitude. And
Well, maybe they did the calculations in a different time zone for this paper.
Ah, there you go.
yeah, right.
That could very well be.
Yeah.
All right, let's move on to our paper today, which continues the arc of new vaccine systems. And last time we talked about making an influenza virus vaccine where you introduce stop codons in the viral genome, you propagate those viruses in suppressor cell lines, and then when you make your virus stock and you infect an animal, it will not replicate. It will enter cells but not replicate, and it induced, as we saw last time, a nice protective immune response. In today's paper is a similar story, but a different technology, but new, and I thought really would be interesting to look at. It was published in Nature Medicine Advanced Online Publication. It's called a chikungunya fever vaccine using an insect-specific virus platform. And the first author is Jesse H. Erasmus, and the last author is Scott C. Weaver. And the authors are from the University of Texas in Galveston, the Baylor College of Medicine, the University of Alabama at Birmingham, and U.S. Army Medical Research Institute of Infectious Diseases, fondly known as USAMRID.
Right.
Right, Dickson?
Right, Vincent.
So here is the story here. A chikungunya, of course, is an alpha virus in the togavirus family. It's a positive-stranded RNA virus. It has a capsid surrounding the RNA, and then around the capsid is an envelope in which is embedded glycoproteins. And chikungunya, as you know, is a virus it's an emerging virus. In fact, emerged probably around the time of Zika, and has spread globally. Causes an arthritic disease, a rash disease, but also with joint arthritis. And as Michael Diamond told us a couple of weeks ago, you can have long-term problems with your joints after infection. So many people are infected. It's transmitted by mosquitoes, and so vaccines are in development. As we'll see, there is a infectious attenuated vaccine that is under development as well, which is made by the old technologies of passing a virus in cells and looking for loss of virulence. Now, the approach here is to use a similar virus that replicates only in insects and does not replicate in vertebrate cells. All right, and the name of it, Kathy, how do you pronounce it?
It's pronounced
ALAT or ILAT?
Yeah.
ALAT?
ALAT. Well, it depends which pronunciation online you listen to, whether the accent's on the first syllable or the second syllable. But it's the name of a city in Israel, the southernmost city. It's on the very northern tip of the Red Sea. And so this virus was originally isolated from mosquitoes in the Negev desert, of which Eilat is a part.
Okay.
That's a destination for scuba diving.
Oh, yeah?
Yes, very much so.
Yeah. Ras Mohammed is very close to that.
EILAT. Okay, isolated from mosquitoes, does not infect vertebrates. And so the idea here is going to be, can we put chikungunya structural proteins on this virus, and then we'll grow it up in insect cells, and it shouldn't replicate in human or mouse cells, and it should be a good vaccine. That's the idea here. All right. So chick is the target, and ALAT. I'm going to call it ALAT.
ALAT.
Is the vector. This grows really well in mosquito cells, so cell culture of mosquito cells, those are routinely available. And they have an infectious DNA clone. Lovely way to call this, by the way, an infectious DNA clone.
Uh-huh.
As opposed to, other ways, which we won't, those which will not be mentioned.
Exactly.
So these viruses, their RNA genome is plus-stranded. They have two open reading frames. The first 2/3 of the genome encodes the non-structural proteins. And these include the RNA polymerase and protease and other proteins that are needed for RNA replication. When the viral RNA gets in the cell, this open reading frame is initially translated. Then there's a stop codon, and downstream there is a promoter for a subgenomic mRNA from which the structural proteins, the capsid and the glycoproteins, are translated. So in order to access the coding region in that subgenomic, in that second open reading frame, the RNA has to be replicated to a minus strand, and then you have to have a subgenomic plus mRNA made from that minus strand. And so what they have done is to substitute the capsid and the glycoprotein coding region of ALAT with that of chikungunya. So they have a recombinant virus, basically. And this virus grows well in mosquito cells. It makes nice plaques, and they show a picture of plaques, just lovely. They do actually solve the structure of the virus and show that it looks very much like ALAT, which was solved before. Is it ALAT or chick? I can't remember now. No differences between the wild type and ALAT chick structures were observed.
It looks like chick.
It looks like chick.
Yeah.
Mm-hmm.
Yeah, it's previously published chick cryo-EM. Of course, you wouldn't expect there to be many differences because you've substituted the structural proteins of chick for those of ALAT. And that's kind of
so
extra information.
Chick on the outside, ALAT on the inside.
Oh, is that like turducken?
Yes, I guess so. I guess it would be kind of a Chick-fil-A.
Wait, chick on the outside. Yeah, ALAT in the... We have to make a title out of that. It's perfect. Turducken. Nathan, you know what turducken is. Yep. He doesn't seem very excited.
Shaking his head reluctantly. Yes, I know.
We've used turducken in titles.
We have. So this looks like a chick, and it replicates well. Looks great. They so then they move to vertebrate cells, and they now remember this has the chick glycoprotein, so it should be okay to bind and get into vertebrate cells. And in fact, they show that it binds to vertebrate cells. They make a fluorescent virus and show that it attaches to cells by flow cytometry. They do electron microscopy, and they can see the virus particles getting in by what looks like the clathrin-dependent endocytic pathway. So it looks like it's getting in, but it doesn't replicate. So they put a reporter into the genome to see if it's replicating. Now, the non-structural proteins seem to be translated, but the genome isn't replicated, which suggests that they don't get structural proteins made, suggesting that replication is not happening because you would need replication to get that subgenomic mRNA made. So this is important in an animal cell. They're using viro cells here. The virus attaches and gets in. A little bit of protein's made, but the genome doesn't replicate, and you don't get infectious virus. I guess that's the most important thing. That's the bottom line there. Whereas, of course, in a mosquito cell, it replicates great and grows to high titers. I'm really jealous of these titers, like 10 to the 10th PFU per mil.
It's a crystallized.
It's huge. It's huge.
How do alpha viruses do that so well?
Grow so well.
Yeah.
Comparing how viruses achieve their titers is really not known.
It's kind of like asking a why question.
Polio, we're always happy with polio, which grows to 10 to the 9th PFU per mil. But this is substantially more, is 90% more, right? That's a lot. And Zika is far less, which is a flavivirus, different family, but kind of the similar structure, an envelope with glycoproteins in it plus stranded RNA.
So, Vincent, nobody knows what the limiting factor is to limit the production of a virus?
No, not really. And why different viruses vary widely in their ability to
each one uses a unique set of host cellular machinery, so maybe it relates to that, no?
Sure, could be.
But I wonder if supply and demand plays a role.
There would certainly be some theoretical upper limits. You can't make more biomass of virus probably than you have biomass of cells, right?
Yeah.
Well, 10 to the 10th, I don't know. Does any virus go higher? Does anyone know? 10 to the 11th? Without, concentration?
Or has anybody...
Well, here, 10 to the 12th, 10 to the 12th, 10 to the 12th.
Has anybody studied hosts, like lentiviruses that grow so slowly but are in long-lived primates?
Not really.
Versus...
No, remember, everybody listening out there, this man is a practicing lawyer. Is that a typical question from a prize? I think not. That was a fabulous question.
Yeah, that's an excellent question. If you have a virus that's infecting N cells in a body, what is its titer?
Mm-hmm.
Yeah.
But people in general don't address that question. Why does this virus grow better than this one? It's really not. We make mutations in genomes and it reduces the yield, and we say, okay, this protein is involved in high yield, but we don't really look at the natural differences.
Yeah, human adenovirus titers get to be pretty high.
Mm-hmm. How much? 10 to the 9th?
10 to the 10th, 10 to the 11th. There is a concentration step in that you spin down the cells and throw away the media, and then you freeze thaw 3 times, and then you take that. So you have concentrated just right off the bat. But yeah.
Now, for some vector preparations, experiments where you're injecting vectors into animals or even people to deliver a gene, then they're typically concentrated. And adeno-associated virus, sometimes you can get 10 to the 12th PFU per mil, but that's because you're concentrating it.
Right.
You really want to put a lot of virus into the...
Yeah, you can buy adenovirus. I'm looking here online, 10 to the 12th PFU per mil.
I'm sure it's concentrated, right?
So I also wondered, in the virus world, how many viruses produce inclusion bodies, which may turn out to be crystallized pure virus? Is that possible?
Look at that little circle chart on my desk. Where is it?
It's right here.
I found this thing. I was cleaning out drawers. What year is it from?
You said 1970.
1970s. It's this circular thing that you rotate, and it's got a dozen viruses on the outside. Right. And you pick your virus, and then little windows slide over information on the chart on what cells you should grow it in, how you can tell if the virus is growing, how to titrate infectivity.
Right. Oh, it's a slide rule.
Yeah, it's circular.
Or a color chart, a color wheel.
It's so funny because there are only a dozen viruses, like polio and vaccinia, adenovirus, and others, influenza. And it's just so simplistic now. Here it is 45 years ago. So, Dickson, just look at that. It talks about inclusion bodies.
I'm looking. And the other thing, I only see one virus here that actually has an inclusion body that's well known. That's rabies. So
Negri bodies.
But adenoviruses do too.
Mm-hmm. What are they called?
What are they?
Kathy,
they're called inclusion bodies.
Just inclusion. Because sometimes, they're named after the person who discovered them.
Yeah.
Right.
Like Negri bodies.
Do we know what they are? That's the point.
Yeah, they're usually replication complexes.
I see.
Well, for adenoviruses, we think they're viruses.
Viruses. Right. Yeah, they can be mature viruses. They could be viral factories, right, where they're being assembled. So it's a combination of assembling viruses and mature viruses.
Isn't that the best way to package something up in the smallest amount of space so you get lots of something?
It's probably efficient to have all the components in one place, yeah.
Yeah.
Hexagonal closest packing or cubic closest
Yeah, that's
packing.
right. That's right.
So in this case, I just want to clarify. So these recombinant viruses attach to mammalian cells. They get in, they make non-structural proteins, but they don't get a functional replication complex because this, and they do that by trying to measure translation of the subgenomic mRNA, and they get very little of that. So they're assuming that they're not getting replication complexes. So next they go into animals. They use a mouse model to ask what these viruses are doing. And for comparison, they have a infectious attenuated virus, and this has been produced by multiple passages in MRC5 cells and 18 plaque-to-plaque passages. And this virus, which was done, in the 1980s, I believe, and apparently this is a candidate, 1986. This is a candidate human vaccine, which is going into clinical trial. So that's one of their controls that they use here. So the first thing they do is infect mice. These are wild-type mice intracerebrally, and they measure viral loads of virus in the brain and also survival. And this vaccine candidate, we'll call it 181 clone 25, they get brain inflammation, and one of the 6 mice die. But the recombinant ALAT virus, ALAT chick recombinant, no signs of any disease, no lesions in the brain. Now you may ask, why is this a vaccine candidate, the 181 clone 25, if it causes neuropathology? Oh, they're putting the virus right in the brain.
Right.
So, if you do that with the polio vaccine strains, you will also get some pathology as well.
I'm trying to find it, but I thought I remembered reading that there are already some known side effects of the 181 clone 25 in people.
That's absolutely right. There are some side effects. Yeah, where is that? I don't know if it's before this. I think it's part of the discussion where they're talking about why this is why their system is better.
Right.
Where is that?
They're probably shouting at the podcast now. It's right there. But at least we saw it somewhere.
Yeah, that's right.
We'll find it.
Somewhere in the paper is this mentioned.
We'll find it.
Oh, yeah, it's right at the beginning of the discussion. Human phase 2 trial. People developed arthralgia after vaccination with chick vaccine 181 clone 25.
Yeah, that's not good because that's the disease you're trying to prevent.
Right.
Right.
Yeah.
Right.
Arthralgia. Then they used the same experiment, intracranial inoculation. They used interferon type 1 receptor knockout mice, which are going to be much, much more susceptible because they don't have an interferon response. In this case, all 6 of the mice infected with the vaccine candidate 181 die within 2 days, and the recombinant inoculated mice survive for 120 days. So they also passage it 5 times from mouse to mouse, IC, mouse to mouse, and this recombinant virus, all the mice survive. They don't detect any viruses after passage 1 or 2. So it looks to be genetically stable. In other words, you're not selecting for viruses that are able to replicate in mice by this passage.
There was no mouse-to-mouse resuscitation of the virus.
Alan, I resisted.
You were going to say
held
that?
off. I was going to catch the ire of Vincent, but I'm so glad you beat me to that one.
Then the last set of experiments, they asked, do you get an immune response, and is it protective? So they immunize mice with these 2 different viruses that we're talking about. The recombinant virus induces seroconversion very quickly, 4 days, faster than the other virus. And they measure neutralizing titers. Of course, the controls remain seronegative. They also look at T-cell responses. They see an increase in CD8 positive T cells, but not CD4 positive T cells. And these are virus specific because they're using virus peptides to look for those as well. They also have a formalin inactivated version of the recombinant virus, which I forgot to tell you. If you this virus, again, you treat with formalin to inactivate infectivity. That will actually not attach to cells. So the formalin inactivation is blocking attachment. They get a far less strong immune response to that, which suggests that the uptake of the infectious recombinant virus maybe into antigen-presenting cells like dendritic cells or macrophages. Maybe that's important for the good immune response. And we suggested that last time for the influenza virus made in the stop codon suppressing cells. The immune response to that was much better than the inactivated influenza vaccine, maybe because it's getting into cells. This immunization provides long-term neutralizing titers. They go out 292 days post-vaccination, almost a year. Wow, this work was done a long time ago. Mm-hmm. And they're still fully protected. Actually, they're challenged. This is part of the challenge experiments. Yes, sir.
I had one complaint with Figure 3 where they're showing the immune response. And they have what appears to be a robust immune response, at least I assume so, because there are no labels on any of the axes of any of the graphs.
Figure 3? Really?
On my copy of the paper, I've got no labels on the graphs.
No, they're here.
Oh, yeah, they're here.
I'm sorry.
Really?
Yep.
You must have got a bad copy.
On a hard copy and online, yeah.
I got a bad PDF.
Where did you get it from?
Off the Nature
site?
Actually, I guess I downloaded this from the press site, which may have been an early version of it. Maybe that's the problem. Okay, disregard.
Yeah.
Looks good. Yeah, the labels are all there.
All right.
Yep. So these immunized mice, they're challenged, and they do it in short term, 30 days post-vaccination, or 292 days. And all the mice challenged with either the vaccine candidate 181 or their recombinant virus, they all are protected from viremia. The recombinant virus infected mice are protected against foot pad swelling, whereas the other mice are not. This is not a lethal challenge model. And this lasts for a long time, 292 days. So this is one dose, and it lasts a long time. They also
bear in mind, mice only live a couple of years, best case. Correct. So this is like years later that the challenge is.
Yeah,
these are middle-aged mice getting this challenge.
I was going to say, in people years, that's a long time.
Yes,
right.
So then they also did a challenge in A129 mice, which lack the alpha-beta interferon receptor. And in these mice, infection with chick is lethal. And again, this is lethality is prevented by immunization with the recombinant virus. And so is immunization with the human candidate strain 181 clone 25. They're protected from death. But, of course, that, as we said, has some issues in people, so it may not be ideal. And then they do a non-human primate experiment. They vaccinate 5 male macaques, 3 to 5 years old, and then challenge them. And it's really interesting. They note that they've implanted these with a telemetric device to measure temperature at 1-minute intervals.
This is cool.
I suppose you could sit home with your smartphone and monitor their temperatures, right?
Well, what's really cool about this is that you see the diurnal cycling of the body temperature.
Yeah, that's right.
And so it's an extremely sensitive assay, because if you're just checking temperatures once a day, and of course, you can't just hand them a thermometer and tell them to put it in your mouth. You've got to do this the hard way. So there's the invasiveness and repetitiveness of having to check it every day, but then also you might miss some short-term fever spike. Mm-hmm. And with this, you've got this incredible detail on it. It's a really nice system.
So it goes from about 38.5 to 35 or so, 35.5. Yeah. That's the cycling. So they immunize some with the recombinant vaccine, then they have a mock vaccinated control. And the mock control after challenge, you can see a spike in the temperature up to 40 degrees. Mm-hmm. And the vaccinated subjects have normal temperatures, and they find no viremia in those animals. So vaccination prevents viremia.
And in fact, that spike that you see in the unvaccinated one illustrates the importance of this telemetric monitoring, because the spike is just like a few hours.
Yep. Mm-hmm. Yeah, you would miss it. That's right.
Isn't it 2 days, Alan?
It's at the 2-day point. It starts at about
Oh, right.
one
But it only
day
lasts.
point.
It lasts a few hours.
Yeah,
Yeah.
it's
Right.
over by the end of day 2.
Right.
That's pretty cool. I'd like to have an implanted telemetric device in myself.
How do you know you don't have one?
Well, you could. I mean, it's moving toward that with the smart watches and
Yeah.
such. You could certainly measure body temperature. Could probably get pulse ox and some other data out of that.
Well, they could have just given the non-human primates an Apple Watch, right?
Yes, of course.
And they have a device that constantly measures blood sugar,
Mm-hmm. Yeah.
and
Yeah.
that's quite remarkable.
That's with
You
the
can
watch
also
also,
have-
right?
Yeah.
But you can also have, for animals, measure blood pressure, and there's lots of
Lots of things, yeah.
telemetry things. Yeah.
Very cool. Yeah, things are very different from the old days, Dickson.
Also,
Yeah.
they get a lot of these ideas from Star Trek. You know that, right?
Yeah.
I'm not joking. They're trying very hard to come up with the bones scanner to give you
Oh, yeah,
the health
for sure.
You
parameters.
cannot talk to anybody in the medical device industry for more than 10 minutes without them mentioning tricorders.
Exactly.
Really.
Exactly. No kidding.
But this chart is probably the closest thing I've seen to something like that. I mean,
Yes.
it's really.
And of course, they communicate with their Dick Tracy watches.
Right.
In the last experiment, they show that antibodies generated in these non-human primates will neutralize several chick strains. So it has the capacity to neutralize strains from Asian, West African, and Indian Ocean lineages. It's a single serotype, but there is some variation. But these antibodies neutralize all, despite the fact that they're made with one of the isolates. So that's the story, and it really is similar to last week. The idea is that you can make an infectious vaccine. It will replicate in the right cell, in this case an insect cell, but then you grow up your vaccine, you put it in a vertebrate host, in this case mice or non-human primates. It replicates, it infects cells, it doesn't produce new viruses, but it makes a pretty good protective antibody response, or immune response, I should
How
say.
about that?
So I like it. I think it's very neat. Two different
Yes.
approaches. Again, we don't know if this is going to be better than existing vaccines. Of course, for chick, there is none. But if you want to make an infectious vaccine, this is very safe, right? Because this is
Looks
not
like
likely
it.
to revert.
Yeah.
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 What is the vaccine that they've recently developed for Ebola?
What do you mean the vaccine they've recently developed? You mean the one that you saw on Yahoo News today?
No, the one that I heard Tony Fauci talking about on NPR this morning. It's been tested in people, I thought.
Well, we talked about that on TWiV. You know, if you listen to the podcast, you would know that, wouldn't you? So there was a VSV Ebola recombinant that was tested in West Africa.
Right.
And until the point where the outbreak fizzled out, and then they
Right.
couldn't test it
Right.
anymore. And it looked
Right.
pretty good. We talked about it on TWiV. Do you remember, Dickson?
Of course I
You go-
did. But Tony brought it up to date
When did Tony talk?
this morning.
Yeah, maybe he was talking about that. Let me see if I can find it here.
It was a vaccine that
Meanwhile-
you can give to people after they're exposed to the Ebola virus.
What do you want to do?
Well, that's
Kathy?
the idea anyway. Go ahead.
Exactly.
Well, if it's, I was going to tell you a little about some of the investigators on this paper because I know one of them and I did
yes.
some research on others.
Go ahead.
So
Go ahead.
the senior author is Scott Weaver, and Scott was a graduate student in the same lab where I had been a graduate student, but Scott's quite a bit younger than I am, so we did not overlap. And there's an additional Holland lab connection. So this was in John Holland's lab where we were both students. An additional Holland lab connection in that someone who was a postdoc at the same time as Scott was there as David Clark, he was working in industry and had someone working for him, Farouk Nazar, and David Clark sent Farouk to Scott's lab, and in fact, Scott has been the beneficiary of at least 3 people by that same pipeline. Farouk was the first author on the first publication of the ALAT virus, on which Ian Lipkin is also a co-author. I don't know if you knew that.
Nope.
From 2012. And so that's a little bit of background about Scott, who's quite active in all kinds of things, emerging viruses, Zika. So I can't remember if we talked about his animal model for Zika and so forth, but that's background on Scott at UTMB. And then the student is Jesse. And Jesse grew up in South Africa and then had a case of bad viral encephalitis. Oof. And he said, I think ever since then, I've had an infectious disease interest in the back of my mind. Pun intended.
Dear.
He came to the U.S. for middle and high school and went to college, focused on an engineering degree, but then he decided maybe he would go to medical school, and he worked in Mario Capecchi's lab at the University of Utah, and then realized that he loved research, so he scrapped the medical school idea and applied to graduate school and ended up in Scott's lab. And he's now a postdoc in Seattle at a research institute, the Infectious Disease Research Institute, where he's working on developing new RNA-based vaccine platforms for emerging infectious diseases. And he hopes to be able to continue the ALAT virus platform as part of that. So then I asked him if there were special contributions by others, and he said definitely from Farouk Nazar, who I think the two of them overlapped for 2 years, and he did most of the groundwork characterizing the ALAT virus and did a lot of dedicated time training Jesse in how to do this work. Jesse also went to Ilya Frolov's lab to learn how to clone the virus, and so he and a postdoc, Dal-Young Kim, played an important role, and they are co-authors on the paper. And then I said, do you have a strong memory of an especially exciting or interesting day during this research project? And he said, every time I fixed and stained a plaque reduction neutralization test, it was amazing. And they expected originally to have to give 2 doses of the vaccine, and when he did his first PRNT is what they're abbreviated for short, the neutralization test, he thought he was really bad at it, so he repeated it because it was such an astounding result. So, but based on that, they just did all the subsequent studies with single-dose vaccinations. And so he said, pretty much every time he did one of these assays, it was just astounding to see such high antibody titers, even 10 months after a single dose. And he said doing the electron microscopy was the most fun. He got to learn how to do that. I asked him about other key collaborators. So I mentioned Ilya Frolov, but he also mentioned Dr. Tian Wang and her postdoc Huanlei Luo, who helped him do the T-cell assays. Vsevolod Popov and Fred Murphy for the EM work. And the animal resource people at UTMB were really good at that. Cryo-EM, Wachu and Jason Kelber. So that was that structural work that they did. So they had a lot of people in on this paper, and it sounds like he got to learn a lot of the techniques at some level, so that was fun. And I think that's the background I wanted to give you. I only had one scientific question for them, and it isn't something that we talked about in great detail, but in one of the early figures, they are looking at the growth of the virus, and they grow it both in 10% and 0% serum, but they didn't really mention why. Mm-hmm. And I asked him, and he said, well, that's because down the line for production, it might be useful to know that you can get such high titers in the absence of serum. Mm-hmm. So that's what that was about.
Yep.
So
make it a lot cheaper.
Yeah.
Good. Thank you, Kathy. All right. So Dickson, here's what I found out.
What did you find out?
So what Tony was talking about, Tony Fauci, and what's in the Times today is that the final report on this ring vaccination trial, which we originally covered on TWiV earlier this year, which was an interim report, the final report has been published.
Okay.
And that's what saw the 100%
And they say 100% efficacy. And in fact, I think even the interim, they said 100%. We had a lot of criticisms of that because the numbers were so small.
Well, that's what Tony said, too. It was just very low numbers.
Next week, Dickson, we'll take a little look at that because we're going to do a year in review, and I think Ebola is part of that.
Well, okay.
And we'll look at the final report next week.
There you go.
How do you think?
Sounds good.
What do you think about that, Dickson?
I love it,
Kathy?
Vincent.
Is it a plan?
Yes.
And there were like 23 cases, is that right?
Yeah, that sounds like about right. Out of thousands of
23 cases in the delayed vaccination arm and 0 cases in the immediate vaccination arm.
Yeah, so that's
which is good. That's certainly a good result. And given this was on the tail end of the epidemic, you just weren't going to get the kinds of numbers that you'd like. I mean, you don't want to see anybody get sick, but you need to see robust statistical evidence, and they couldn't do a large enough trial under the circumstances. But yeah, it's pretty impressive.
So when I was at Rocky Mountain Labs a couple of weeks ago, I asked several people, would this vaccine be used in the next outbreak? And they said probably, but it's not clear. So they have a stockpile.
Right. They've bought 300,000 doses of it now, apparently.
And, they would probably give those to healthcare workers. And they would do ring vaccination when someone was infected. They would vaccinate their entire family and all potential contacts and so forth.
And then we'll have a much bigger data set.
Yeah. This episode is brought to you by CuriosityStream, the world's first ad-free nonfiction streaming service. They have over 1,500 titles and 600 hours of content. Dickson, if you looked at these movies continuously, 24 hours a day, 7 days a week, it would take you over 2 years to get through them.
And you'd probably be dead.
Or wouldn't remember anything.
Yeah.
But you don't look at it continuously, so it could last a long time. And, of course, they're always adding new content. It was founded by John Hendricks, who was involved in the startup of Discovery Channel, so you're going to get real science. Available in 196 countries, which Dickson assures me that's it for the world. 196 countries. Maybe there's a new one today. Who knows?
Well, it depends on how you count countries.
Well, United Nations countries.
Yes, United Nations count would be 196, but then, they count Taiwan as part of China.
I hear you.
Wow.
Yeah.
So what they have is science and technology, documentaries, nature, history, interviews also, lectures. They don't have my lectures, but they have other people's lectures. I should tell them to put my lectures in.
Absolutely.
Some examples. Digits. It's a 3-part series hosted by the creator of Veritasium, which is a YouTube science channel, very popular, Derek Mueller. 3-parter, which explores online safety and security, includes interviews with former NSA contractor Edward Snowden and Vint Cerf, co-founder of the Internet, along with Al Gore. Is that right?
This is true.
Stephen Hawking's favorite places. This is a brand new documentary in which he pilots a computer-generated spaceship and stops at his favorite places. What would your favorite place be, Dickson?
I think I'd pick the Orion Nebula to start
Orion
with.
Nebula. I want to go to Tatooine.
Where?
Tatooine.
Oh, so do I. I meant to say that also.
Do you know where that is?
I haven't got a clue.
Do you know what it is?
I have no idea.
Yeah.
Fictional?
It's fictional.
Tatooine. That's probably why I don't want to go there. I have enough trouble with reality.
They have a 3-part series on the
It's warm there.
the 14 billion year history of the universe. That takes a long time to watch. And underwater wonders of the national parks. But if you just go to curiositystream.com, you can search what they have, and you don't even have to log in. Let's just do it now. Dickson, give me a search term. Should I search Helminth?
Sure.
I don't think that's going to come up.
Yeah, no, it will. I'm sure it will.
You think so?
Yep.
There are helmets everywhere.
There are.
Let's see. How do I do? Oh, they changed it.
If it comes up, you'll have to call CuriosityStream and let them know they have a worm on their site.
That's exactly right. And a wormhole is another place I'd like to go, by the way.
I put a new Wi-Fi network in my home, and it's called Viruses, so nobody will sign into it.
Right.
Hey, I've had that forever.
Very good.
You have?
My Wi-Fi network? No, actually, that's not true. It's my computers all have virus in their name.
Really?
So in the university networks,
No, nothing for Helminth, Dickson.
Nothing for.
Let's try worm. One result: the worm hunters,
Well,
armed with spades, GPS worm locators. Oh, stop playing! Don't stop playing. They're looking for super worms. Well, that could be. I would look at this. I don't know what it's about, but it's called the Worm Hunters.
super worm.
It's pretty neat. Anyway, this is cool stuff. They also have one of the biggest super high definition libraries. That's 4K content over 50 hours. And how do you get this? All right, they have monthly and annual plans available. They start at just $2.99 a month, which here in New York City wouldn't even buy you a cup of coffee, I think.
This is true.
How much is it
Buy you a
downstairs?
parking place.
How much is it downstairs, Dickson, a cup of coffee?
It's actually $2 and some change.
Yeah, it's because it subsidizes by Ardeen, right?
I don't know that for sure. I'm not about to agree to that.
Columbia subsidizing something for the people who work there?
So.
No.
It's a joke.
No,
It's a joke.
It wouldn't happen.
It's a joke. Check out curiositystream.com/microbe. Use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. As far as I can calculate, that is 2 entire months free of one of the largest nonfiction 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign-up. We thank CuriosityStream for their infinite support of TWiV because the ads are there forever, as Stephen said, and we talked about earlier. Right? All right, let's do some email. All right, the first one is from Paul, who writes, TWiVlers. That is one I haven't heard before. TWiVlers. 13C in Philadelphia with very high variance and low clouds. Further to the conversation about authors, they has a PhD comic on the number of authors per paper per field.
This is good.
This is wonderful. So the least number of authors are in the fields of feminisms and women's studies, ethnic and cultural studies, language and linguistics, 2 or 3 authors. And then, all the biological, immunology, genetics, neurology, 20 authors, 226 authors in high energy and nuclear physics.
Yeah, I was going to say physics.
Why even list the authors they write?
That's right.
But even the little tags that they up at the 1 and 2 authors, the lone wolves, and then serious trust issues for the 2 and 3 authors, included others mostly out of politics, and it's just very, it's fun.
It's good stuff. Thank you, Paul. One more. So we have a leftover limerick contest.
Right.
Michael writes, I love TWiB, TWiM, and TWiV. I guess he means TWIP, right?
Don't know.
We don't do a TWiB, so it must be TWiP.
Well, okay.
I grew up in a medical family. It skipped a generation because I turned down Harvard or Yale for a music education. My mom was a nurse. My son is
Ing.
I don't know what that
One.
Intern?
maybe. One of you has a limerick contest, most likely expired, but here's my entry. There once was a virulent virus which tried with all might to retire us. We know it's a pest, but hardly the best. I'm sure that you know Miley Cyrus.
I
don't get it.
Well,
You get it, and I know. It's amazing that you would get it.
have you ever heard Miley Cyrus sing?
I guess.
I don't think you have, because if you had...
Didn't she do something about a sledgehammer?
There's something... She has a fabulous
Wrecking
voice.
Wrecking ball, right.
She's got an amazing voice. I mean, she's in the news all the time because she's kind of gone hyperbolic with regards to sociological events of various sorts, but her voice is amazing.
She has more money than you do, Dickson.
She'll have more money than most of us. She's really quite a talented person.
All right. Well, I guess he doesn't like Miley Cyrus. Alan, can you take the next one?
Sure. Hannah writes, oh boy, I'm going to butcher this.
let
Bonjour à tous francophones et TWiVnauts.
Yeah, it's good.
Okay.
Aha.
And sends a pic, which I guess we can do at the end.
Sure.
And then public message. Hello from FEMA Region 5, Midwest. Kathy is also in FEMA Region 5, I believe. It's 13 degrees, sunny, and the air is crisp, and my cat is meowing. How are you to all? Thank you for your courage, production, desire to teach, consistency, and communication. Learning the opinions of others, especially if the opinions and people can bring something new for me to observe, has always been a tryst into absolute bliss.
Trip?
A trip, maybe?
Tryst, maybe. T-R-Y-S-T?
Yeah, that would be...
Okay.
It's interesting that didn't get highlighted by the spell check. Realizing I knew nothing of your topics but wanted to learn, the natural choice was to go along for the ride and inform myself as new things cropped up. Months of listening later, still clueless at times, I feel the ability to locate learning resources growing. Thanks to you, I'll be returning to schooling with an obsession. I, as well, will be looking forward to an insect podcast. As will I. That's a great idea.
Vincent, you weren't in on the conversation. I sent it to Rich and Alan and Sarah to listen to, and Alan came up with the better show title, which was
Ants in Her Plants.
Oof.
Should have been your previous TWiVo, but all right. Continuing with Hannah Lore's letter, perhaps you'd have luck goading Dr. Despommier into recording urban agriculture episodes while fishing. All my media comes from books, articles, and podcasts. I cut the cord before turning 18, 10 years ago.
Wow.
Okay. Podcasts, OPML. So this is a list of
all the podcasts she listens to.
Of all the podcasts she listens to.
Yeah, a lot of them.
In
wow.
In XML format.
Yeah,
wow.
that's cool. So you could just click on either. Yeah.
Wow.
We're there. We got all the TWiVos.
Yes.
Hey, look, there's a This Week in Drugs. I didn't know that.
Oh,
hey.
yeah, man.
Okay. The Vintage Space Podcast. Wow.
And a lot of computer related stuff. She's in systems admin, I guess.
Yep.
And then with the utmost admiration, Hannelore S. In reply to Steve's questions on TWiV 415, to filter what's presented on the web, a bit of aid comes from Web of Trust to flag links in conjunction with uBlock Origin, script safe, HTTPS everywhere. My browsing is a breeze. To cut back on Facebook ads, I only browsed via mbasic.facebook.com and other minimal mobile versions that didn't do Ajax or other fancy web bugs web content. WOT puts a color-coded marker by the link. Script safe acts like no script, and only allowing HTTPS via HTTPS everywhere prevents some nasty frames from loading. For more, look at this handy site, and it's prismbreak.org, which is also the name of a video game character, but that's opt out of global data surveillance programs. Cool.
And it's prism-break.org.
Yes, prism-break.org.
So Prism is the NSA thing, right, that grabs everything?
Right.
Okay, cool. Wow.
So some
a lot
good
of
tips and
security
flavors there.
stuff here.
Yes.
Okay. Well, thank
All right.
you, Hannelore. Kathy, I think you're next, right?
Mm-hmm. Anthony writes, some 15 years ago, Esther Dyson said that with the rise of the internet, the price of information will tend to zero. Profit is to be drawn from events and products. Ms. Dyson did hold the New York Times up as a possible rule-proving exception. This appears not to be so. With the New York Times promoting its store and events, the paper indeed is, as Esther Dyson predicted, following the example of the Grateful Dead, for what it's worth. And then there's a link to an article from Wired, July 1995, Intellectual Value by Esther Dyson. What happens to intellectual property when it gets on the internet?
This is true. This is certainly true, isn't it?
Yeah.
Everybody expects everything to be free.
Yeah.
So the Grateful Dead used to give away, recordings of their concerts.
Right.
Or they
let you record it.
They let you record.
They let you record.
Their own bootlegs there.
And people distributed those bootlegs, and the idea was people would then buy their music, right?
Yeah.
Right.
Right.
So
Will.i.am did
go to
more
their
importantly, they would
concerts.
buy tickets to the shows, which was really the experience of them.
Exactly.
I understand.
Exactly.
Yeah.
By the way, did I already tell the one about what the Deadhead said after he stopped smoking pot?
Mm-mm.
Man, this music sucks.
Yeah, I've heard that. Yeah.
We're going to get letters on that.
I guess.
Nathan, do you have any thoughts about what happens to intellectual property when it gets on the internet?
Well, I think it's definitely hard. Although I think they're interesting business models like advertising on YouTube and things like that can return revenue to musicians and other things like that.
Yeah.
I do think it's part of the difficulties facing our new press and news and compensating, sort of journalists across the board, unfortunately.
Well, and I think a lot of the problem in the media industry is not intellectual property. It's that for many, many years, the business model of news has been selling a commodity, which is the information that's in the newspaper. And if you go, I mean, I've had this experience where I'll see some news story and click on the link and it pops up some sort of ad blocker or something, or the site is a mess, or it directs me to the wrong page, and I'll just Google the topic and there'll be 8 other sources covering exactly the same story. And under those circumstances, it's not that you don't have intellectual property on the story itself, because that's just what's happening. And you'll just pick whatever the first link is on telling that story, whether it's the New York Times or Joe Schmo's blog, it doesn't matter because you're still going to get the information. And what people are after is the basic information, which has gotten very, very hard to sell at a profit.
Yeah, I was at a Seoul Digital Forum meeting in 2009, and at this meeting were two protagonists. One was Sumner Redstone, who was at that point in charge of CBS Records, I believe, and all kinds of other things to CBS, or was it RCA? I'm not sure which company, but it was a very, very large record company, and they managed the Red Hot Chili Peppers music. And, they get paid a lot of money up front to record for them. And at the same meeting was will.i.am, who is one of the lead singers for the Red Hot Chili Peppers. And they stood on the stage and toe-to-toe went off at each other as to why the record companies need the musicians to stop putting their music up online. And they said, the reason why we do it is so that people will know what we're doing and they can show up for our concerts and we can make money that way. And somebody said, well, didn't you make enough already with the contract that you signed with us? And it just kept going back and forth between these two people. And everybody at the end of it was thoroughly confused as to how the music industry actually works.
Everybody is confused, including in the music industry, as to how the music industry works.
I agree.
But it is true that tours tend to bring in a lot more money, have always brought in more money than the albums. The albums have always been a mechanism for selling the tour.
Right.
So, yeah, I mean, will.i.am, I think, had a point. They do want their music to be out there, and so they want people to buy tickets to the shows.
Exactly.
With CBS, Dickson.
Thank you.
Majority owner, CBS and Viacom.
Yeah.
Dickson, can you take the next one from Sarah, please?
I can. Sarah writes, Dear TWiV, I have a question regarding the definition of a zoonotic virus. I am a veterinary surgeon with a PhD in molecular virology from Imperial College London, and, of course, am an avid TWiV fan, having seen live recordings at ASV and UKSGM. Having published studies looking at zoonotic viruses in dogs, I thought I had the definition of zoonosis sorted. However, I am now teaching a bunch of bright second-year medical students at the University of Cambridge who have made me realize the existing definition from WHO. A zoonosis is naturally transmissible from vertebrate animals to humans and vice versa, lacks some details. Some studies report that 60% of the viruses have arisen in an animal host and are therefore considered zoonotic, but many of these viruses gain the ability to transmit directly between humans, and then the vertebrate host becomes irrelevant. In the truest sense of the word, I appreciate that viruses such as Ebola and HIV are zoonotic, but is there a point in virus evolution when this terminology becomes redundant or / inappropriate? And when might you say that is? Many thanks in advance for your thoughts, and thank you for the ongoing virology enthusiasm. Sarah.
Well, I would say HIV is not a zoonotic virus at the moment because a zoonotic infection requires fresh virus from the animal reservoir into people, like Ebola, right? And MERS. MERS probably does a little bit of human-human transmission, but it's not very good. So the point at which it's no longer zoonotic is when it's efficiently transmitted amongst people and it doesn't need the animal reservoir anymore.
Yeah.
What do you think?
Right.
Makes sense. Is that good?
And in fact, it wasn't even called SIV once it got into people. It was called HIV.
That's right.
Because H stands for,
human.
I thought there was a history. Wasn't it something about the history where it came from originally?
Yes.
Sure.
I mean,
Beatrice Hahn, a whole bunch of stuff on
Yeah, so, there have been multiple spillovers of SIV into people. And then we have HIV-1, the 4 main groups or 4 separate spillovers, and then HIV-2, there have been even more spillovers for HIV-2. But once they adapt and go from person to person without the need for a new introduction from the animal reservoir, then it's no longer a zoonosis.
Right.
Vincent,
Yes.
do I remember you saying in one of your class lectures that it's your opinion that probably all viruses originally came from an animal? Okay.
Yeah,
Well, if you go back far enough,
that's right. Go back
we
far
know
enough.
We came
humans
from
then.
animals.
Yeah, we came.
Yeah.
Yeah, I think they're all at some point.
But interestingly enough,
Well,
not-
but there must have been viruses that we had when we first. I mean, you'd have to pick an arbitrary point to say here is the first modern human, and that's a separate problem. But I'm absolutely certain that first modern human had viruses.
oh
Yeah,
yeah,
it probably got viruses from its simian antecedents, right?
Right.
There you go.
Yeah, but at some point those came from
And those
the animal.
became
well, but
human
those
viruses.
are not humans, right? So
Yeah, what's interesting is that I think, from my perspective, is my limited perspective, I must add, is that certain zoonotic infections, like SARS, for instance, did not become a human-to-human transmission epidemic like it might have been feared it was going to, and it hasn't actually come back, as far as I know. But others, like Ebola, keeps reemerging, and so maybe the transmission cycle is more amenable to a zoonotic transmission cycle from that perspective. Maybe bushmeat or something else that also facilitated SIV to HIV. Who knows? But SARS, certainly not.
Right. Well, it's a lot of it has to do with the human factors surrounding how they emerged.
Absolutely.
And SARS emerged in a specific place and time, and that place has now taken measures to prevent it from reemerging. And there are governments in that region that are capable of doing that, whereas Ebola viruses are in a very wide geographic area, spanning places where there are not functional governments in place to keep it from reemerging.
Yeah.
But in places that have effective public health programs where they're aware of this, and yes, we can get spillovers of Ebola virus, when they take proper measures to deal with that, you get one case, and that's it. And they show up at the hospital and they say, "Oh, it's Ebola," and everybody gowns up, and it doesn't spread to anybody else.
I mean, that's what's happened in U.S. and Europe.
Right.
Yeah.
all right, next one is from Heather, who writes, "Hi, fellas. Although I am as far away from the medical field as you can get, I really enjoy listening to your podcast. I am in Toowoomba, Queensland, Australia, and really love the stimulation of learning something new every day. As I left school, having flunked year 10 level, I was 16, and no direction of what I wanted to do, I surprise myself constantly on how much I understand, although mental pictures might not quite match the actual of what you guys discuss. I don't have much patience for novels or movies, but I like nonfiction, factual podcasts that can teach me something I don't know. You all have such lovely radio voices that make listening so easy and enjoyable. Don't listen to the complainers about the format of the show and letters you read out, as it is just what is required for an unscientific person such as myself. In fact, it is the perfect fit. I am guessing that 56 years now of life experiences make up for what I didn't learn in school those years ago. I'm gradually catching up on all your podcasts from the beginning, and I'm up to 134 and enjoying the experience. I have listened when doing my bookkeeping course, but that doesn't work well as I have to concentrate. So I usually listen when I'm relaxing at night to mask the sound of the tinnitus, which I have had all my life, and learn at the same time. The best guess is that I got the tinnitus from a severe dose of chickenpox when 18 months, hence no memory of not hearing this infernal noise. I get very upset when I hear of parents having chickenpox virus parties to get the sickness over and done with for their children. They have no idea that they are possibly ruining their children with future medical conditions, with shingles and other conditions. I'm at the point that I want hearing aids to mask the noise and help hear what people are saying around me, not because of deafness, but the level of the noise in my head. While violence is not good normally, I would love to give these dumb parents a good slap. It is a great point, which we often make, that it's not a good idea because there are side effects of having natural infections, which can be severe, as
we have
you said.
vaccines.
In an earlier TWiV, you mentioned the journalist that investigated Dr. Wakefield in a podcast interview. Some of you, it seemed, I'd really be interested in hearing it if you could put a link on your website. Also, I'm curious as to what year U.S. students start learning physics and biology. I get the impression that it is a lot earlier than here in Australia, where it is the upper 1 or 2 years of school just before going to university. I think college. I think university sounds a lot better and more grown up. Well, there is a difference, though. Keep up the excellent communication work for your devoted followers. So
most U.S. students start learning physics and biology never.
I was going to say the same thing. You know that?
Oh, you guys are cruel.
I was going to say the same thing. They do start learning it in before 8th grade, before 9th grade, before high school, right?
You get a touch.
My 8th grade science class was physical sciences.
Yeah.
Right.
Yeah.
High school.
Yeah, they do. They touch on aspects of it. I mean, my daughter's in elementary school. She's in 5th grade now, and she's had some physics and some biology and some teensy bit of chemistry. So they do try to introduce basic concepts early on.
And then a full course named biology or physics. Biology I had in 9th grade, and physics I had in 11th grade.
Yep.
Biology I had in 9th or 10th grade. Physics I had never in high school.
Gee.
You missed the chance to see all those boring movies. It's the only time I think I fell asleep in high school.
That was one of my best. I loved the course that we had in physics in my high school, and because the teacher was so engaging, and his name was Mr. Greensick. He was also the timekeeper for the high school basketball games, but he had a wonderful sense of humor. And one day, one of the students was acting up or something during the lab and leaned out the window, and he says, "I tell you what. Why don't you continue to lean out that window until you start to fall and see if you can repeal the law of gravity?" And, of course, we all thought that was funny. But, of course, if this kid was psychotic, he might have actually done that, and that would have been a bad thing. He didn't.
Good.
Alan, can you take Trudy's, please?
Yes. Trudy writes, "Dear TWiVers." Oh, by the way, so briandeer.com is the site to go to get that podcast. I don't know if we covered that.
Nope.
Trudy writes, "Dear TWiVers, I have a burning question that I have been dying to ask you for a while. I'm a huge proponent of the flu vaccine, and every year when I get it around mid-October, I announce it on Facebook in an attempt to raise awareness and hopefully motivate others to do the same. Good. I do the same thing myself, except on Twitter. Every time, without fail, I get those one or two comments about how, quote, every single time I got the flu shot, I got the worst flu of my life, so never again, end quote. These are not necessarily comments by people who believe that it was the flu shot itself that caused the flu. I know how to address those comments. Instead, these people truly believe that not only did the vaccine not prevent disease, but even exacerbated it. My own mother, who takes care of my children, refuses to get the flu shot due to this alleged experience. A few years ago, one person even cited a couple of papers to support his position and provides links. Although I haven't read the papers in detail, the data look mostly correlative to me. One of the papers attempts to attribute the increased risk of disease after vaccination to lack of heterosubtypic immunity, but I still don't understand how that would contribute to enhanced illness. The papers do alert to some of the study flaws, such as the bias created by the observational nature of the studies, as well as the lack of randomization. Although one of the studies attempted to validate the disease case definition on the basis of H1N1 seropositive individuals, they admit that participation in the serologic survey was self-selected and the sample size was small. It is also speculated that Canadian Aboriginals, a population featured in one of the studies, are genetically more susceptible to severe H1N1 disease. I do understand skepticism, and I think that's a healthy way to evaluate scientific data, but I remain unconvinced that the flu shot causes enhanced disease. How do I respond to someone who swears that to be the case, especially if they offer their own anecdotal evidence? On a side note, on TWiV 417, Vincent asked listeners which other podcasts they listen to. In addition to TWiV, TWiM, TWIP, and TWiVo, I also listen to Mark Crislip's Infectious Disease Podcast, an intellectual property podcast called IP Fridays, and a financial podcast called Good Financial Sense, Sense with a C. I have a long commute and tend to alternate between podcasts and audiobooks. Every Sunday, I download all the new podcasts, and first thing Monday morning, starting with TWiV, of course, I catch up on all my podcasts. Sometime around Wednesday, when I'm caught up, I switch back to whichever audiobook I'm currently listening to. That is a long commute. Yeah. Anyway, thanks for all you do. So Vincent, you were looking at these papers.
So they are certainly, as Trudy says, they're correlations only. In fact, one of the papers says the finding of an association between prior receipt of the 2008-9 inactivated trivalent inactivated vaccine and increased risk of pandemic H1N1 illness is not conclusive and needs to be investigated further. There can be confounding factors. Maybe the population gets more serious flu for other reasons. I sent out a couple of emails, and I got one response to flu virologists. I got a response from Adolfo Garcia Sastre at Mount Sinai. So the most probable explanation is that natural infection induces better cross-protection against shifted strains. In other words, seasonal vaccine protects you from infection with seasonal viruses, but not against pandemic or antigenically not matched viruses. So when a pandemic virus comes, a previously vaccinated person is not protected, while a person previously not vaccinated might have been infected naturally, and this infection is cross-protective to some extent. at least during a few years after infection. This is why I favor live vaccines, more cross-protective when they work, and also why we need a better universal vaccine. So, he's assuming that it's true, but I'm not convinced that it's true yet. But if it were true, that might be an explanation of it.
Right. I don't buy it.
And also, I think studies in other countries have to be done as well, right? Because that could be an issue. Kathy, can you take the next one, please?
Sure. This is from Ken Stedman. Dear Vincent et al., just wanted to let you all know that the first publication of hopefully many to come from a collaboration that got started in response to TWiV 195. Parenthetically, he says the one with the abysmal audio. I don't remember that. But, anyway, this publication was just published. And so he links to a paper in the Biomed Journal on which he's a co-author. So evidently it's a topic that came up during that TWiV that led to this paper. I just now realized that we should have added TWiV in the acknowledgments. We will make sure that it happens for future papers from this collaboration. Maybe we can get the publisher to add one retroactively. Thanks. Ken.
So his audio was abysmal because he had Skype issues. The network at Portland State was not very good.
TWiV 195 was, they did it in the hot tub. This was the hot springs. That's what Ken does.
Oh, right.
And that's the one your brother, Kathy, wrote in about.
That's right.
Remember that? Because he was shocked at the title. But just joking.
No, right.
All right, Dickson.
Here we go. Sam writes, Dear TWiVcasters, Vincent, Dickson, Kathy, Rich, and Alan, thanks again for a great podcast. I learn more about the fantastic biology at the very smallest scale every time I listen. In addition, your perspective on current topics like Zika is invaluable, although I can recommend Patreon to my fellow listeners as a painless but concrete commitment of support. Regarding other podcasts, I've got quite a few. Other writers have named Freakonomics, or the Frankensteinomics, and You Are Not So Smart, and they're great. In that vein, I also like Planet Money and Point of Inquiry. Chemistry in its element, The Curious Cases of Rutherford and Fry, The Naked Scientists, and Planetary Radio are some examples of scientists covering science. Nature and Science magazines also produce podcasts that showcase research. In order to sustain my mental well-being, I am taking a break from the news for the no doubt uneventful remainder of 2016, but many excellent news TV shows such as the PBS NewsHour and Washington Week in Review are available in podcast form. For lighter listening than current events, I enjoy a couple of horror fiction casts, the H.P. Lovecraft Literary Podcast and Welcome to Night Vale. I'm writing from Tucson, and today is expected to be sunny with a high of 74 F and a low of 48. My app happens to include a flu forecast for sporadic and medium-high pollen index. I know 74 is probably too much to hope for, except for Rich, but I hope it's a nice day when you read this. Sincerely, Sam. P.S. Listener pick? Then we can read that during the picks if you want.
Sure. These are some interesting podcasts that I didn't know about. So I happen to be a big H.P. Lovecraft fan, so I have to check that out. I want to check out the chemistry one, too. That could be cool.
So if you want something less scary than the news, you can listen to horror fiction cast.
Nothing is less
H.P. Lovecraft. Hear about Cthulhu coming to destroy the world.
Oh, Lord.
All right, let's move on to some picks. I think that we've got enough here. We have just a few left, but we can do those next week. Okay, there we go. Hey, Dickson, what have you got for
Well, I have some National Geographic 2016 nature stills or pics, PIC, that are absolutely awesome. They catch nature at the moment that something interesting is happening, and I just got these look staged. I mean, it looks as though that eagle was just put there, and then it's stuffed, and then they put a fish in its talons. To be there at the precise moment that happens is almost impossible, and yet somebody was.
They wait, right?
They wait, and they wait, and they wait.
And they shoot a lot of pictures.
They do, but they've got big chips.
Yes, this is somebody who has thousands of photos of like an eagle tail or something.
Exactly right. But the one I was most perplexed and stunned and then entranced by was the one of the rice fields. It looks as though they're vertical to each other. It's actually a pit. and it starts at the center and works its way.
Oh, yeah,
to the
look at
top.
that. That's cool.
quite remarkable.
Mm-hmm.
Why is there one of Ivanka interspersed among these? Dickson?
You get
have
ads.
no idea. I have no
There
idea.
are ads.
But I must say that those rice paddies, if it ever had a torrential rainstorm come into the area, would soon be a lake.
I love the eye. This is a
Yeah. An alligator or a crocodile, one of those two things.
so cool. Reminds me of the eye in that iguana movie.
Right, exactly
Very
right.
nice. Alan, what do you have?
I have the open access button, which I just found out about. This is a website that's been set up by the open access movement. So it's called openaccessbutton.org. And if you come across an article that you can't get access to because of a paywall, you can paste the URL of that article into this little bar and click Find It. And if it is available somewhere on the web for free, they'll point you to it.
Is this breaking the law?
No.
No.
If it's available legally for
Okay,
free
if it's
somewhere,
legal.
We can get an opinion.
So
on
what
this.
they do, they've got a tailored search engine that is basically searching things like faculty websites and university press release sites and that kind of thing to find all of the papers that are being put out there legitimately for free. And they will correlate your search with that and come back with it. And if it's not available through that route, which I tried a couple of them and they weren't, but probably because they were fairly recent, then you can sign up for free on this site, and they'll, if they can't find a link to it already, they will directly contact the author of the paper. They'll do an automated contact to the author to see if they can get a copy for you.
That's great.
A legal
they will-
copy.
A legal copy sent, a reprint article sent from the corresponding author of the paper.
Yeah, I just checked for today's article, and it says
It's not.
available. You can create a
can-
request. So
Yep.
that brings up this is cool. That brings up an interesting question. How many reprints are you allowed to send people if you're the author on a paper? Is it
can send
unlimited, or is
One per person.
you can?
But then, yeah, I mean, in the olden days, people would write and ask for reprints, and
You'd
Yep.
buy them and mail them, right?
right,
But nowadays they're PDFs.
Yep.
right.
Can you send as many as you get requests?
I think one per person.
I don't think this has been tested in the courts.
I've seen on some sites you can only do 10 or 20. You can only send this out 10 or 20 times, but I haven't seen it everywhere. But I'm just curious. All right. That's a cool site, though. I really like it. It just makes it quicker to find papers that are online. It's neat. I like that very much. Kathy, what do you have?
I picked something that's seasonal for today and for the day that this podcast will be released. It's a diamond radio receiver. It's written about in Science, and there's a little video that if you don't want to listen to the Christmas music, you can just watch the subtitles that are on it. So the researchers manipulated the atoms in a diamond crystal to function as a radio receiver. And basically, they say, where is it? Yeah, the radio receiver is the size of 2 atoms. Anyway, just something very different.
It's cute.
Yep.
I have a little technical quibble with it, but it's not actually a complete receiver. That you have to add a bunch of components to these atoms, and then it's much bigger than 2 atoms. But basically, they've built kind of like an antenna out of 2 atoms, which is pretty cool.
Yeah, that's cool.
Yeah, and they say that it could be good to have in harsh environments. So, I immediately thought of the Martian and Mars and stuff like that.
Right.
I had a technical quibble because every time I go to the page, I have to reload the Science page and try and click on the arrow to get it to play. But eventually it plays. And it's not very long, so it's worth checking out.
Nathan, you have a pick for us?
I do, and over the years I've had other picks, but then I end up getting scooped. Like it's like the Origin of AIDS, or the Parasite Museum in Japan. But this one to the best of my knowledge, hasn't been picked, and that's the Mutter Museum in Philadelphia.
Oh.
And you can go there and you can see sort of Siamese twin skeletons and some 7-foot man and some
soap lady.
The soap lady and some smallpox tissue in formaldehyde and it's a really great thing. And then they also have this, you can buy stuffed animal viruses and bacteria and cells too, which I think has also been a pick of the week here.
Mm-hmm.
I can't remember.
I went to this place several times when I lived in Philly. It's great. And it's
true.
The setting of it, it's not just a Victorian oddities collection. It's actually set in a building that is totally appropriate for that. It's really, really neat.
You expect Vincent Price to welcome you in.
Yes.
Yeah, it's a scary place. You can imagine.
It's a lovely picture of the medicinal plant garden, too. Looks like that's nice
Isn't
to
that close
check
to
out.
not the city hall, but the public health department? Because I remember being asked to give a talk at the public health department, and then I'd say, wait a minute, there's the Mutter Museum right there.
It's downtown.
Yeah. Philly isn't that big. That's true.
No, it's
knowable.
It's a fairly sizable town. It's like the
Reminds me
fourth
of
largest
Boston.
city in America.
Yeah.
It's much bigger than Boston.
But it reminds
Everybody
me of
forgets
Boston.
about it, but it's a huge city.
Everybody forgets about it. Thank you, Nathan.
That's really cool. Yeah.
Yeah.
My pick is an article by Jason Snell in Macworld called How Rogue One, a Star Wars Story, is Connected to the Mac. Now, this sounds a little random, but I have to tell you this story. So it turns out that the chief creative officer at Industrial Light and Magic, which did the special effects in Rogue One, which, by the way, I took my 3 kids to see this week. It's really good.
Oh, good.
John Knoll, Kathy will like this too.
Oh, I know this story. Yes.
And his brother, Thomas was a PhD student at the University of Michigan, and he, at the time in the 80s, wrote an image display application, sort of just to look at images, and he sent it to his brother, and they began expanding the program, and it was released early on, and they called it Photoshop. Now, I remember, so Photoshop was bought by Adobe. They pitched it to Adobe. John Warner was the CEO, and he said after 15 minutes, he said, we got to buy this program. They licensed it, and they released Photoshop 1.0, which I remember. And when you first launched the program, it said written by John and Thomas Knoll. I'll never forget. For so many years, I saw those names over and over and over at the
Still
beginning.
do.
And is it still there?
Well, at least on CS6 it is.
It is.
Thomas Knoll is first, yeah.
Really? Okay. Even at Startup, or do you have to go to the About thing?
No, at Startup.
It's at Startup. And I saw that
started
over
it
and
up
over.
today,
And with
yeah.
subsequent versions, version 2 and 3 and so forth, the list of people got longer and longer and longer, you know. And I think what happened at version 3, something happened at version 3, which I can't find here now. All right, it doesn't really matter. So basically, Noel then is involved with ILM, and he did the special effects, or that company did the special effects, and he's also connected to Photoshop. And the Photoshop connection I thought was cool, and I thought Kathy would like the University of Michigan.
Oh yeah, no,
connection as well.
he's so Thomas Knoll is the brother who did the Photoshop, and there's a Summers Knoll School in Ann Arbor. If you go to Wikipedia, it tells you that Thomas Knoll still lives in Ann Arbor, and I know somebody, I think, who used to teach at Summers Knoll. So, yeah, ever since I moved to Michigan and started seeing the Knoll name in Photoshop when it booted up, yeah.
With version 3, Adobe bought out the Knolls, and Thomas went to work at Adobe, and John stayed on at Industrial Light and Magic. And then at the end it says, it's heady stuff, but how heady can it be really when you and your brother invented Photoshop nearly 30 years ago? So, Noel, last thing here, went on to be the effects supervisor for the prequels. He won an Oscar for Pirates of the Caribbean and was named Chief Creative Officer of ILM in 2013. And he basically got this movie, Rogue One, going, which is really based on the crawl at the beginning, the opening crawl in the first Star Wars movies. You know, it said, a group of rebels have stolen the plans for this killer planet thingy, and they said, let's figure out what happened, and that's where the story comes from. So I think a lot of cool stuff that I remember.
I just have one question for you, Vincent.
Yes, sir.
Why do the Empire's spaceships make noise in space?
Oh, well, spaceships make noise as movies, of course.
Yes.
What's the basis for that?
Well, no basis.
One of the many
there are lots
creative
of,
licenses taken in space films.
Otherwise, it'd be a silent movie.
If they were quiet, it wouldn't be very interesting, But when the ships have all kinds of noise, and when they crash into each other, they make noise. Yeah, it's a problem, Dickson, but
it's a problem.
You can look beyond it. There are lots of other problems as well, but
They don't do that on Star Trek.
They don't make noise? Sure they do.
No, they don't do that on Star Trek.
Oh, I'll bet we have noises on Star Trek.
Not in deep space.
Well, maybe when you're doing hyperspace, not. But I'll check. Are you a Star Trek fan?
I am.
All right, we have a bunch of listener picks. We had Sam, Evolution, Cell Lab, Evolution Sandbox, a cell game for cell phones. And There's a link to this at the Google Play Store.
I watched the video, and it's really fun just to watch the video of the game.
Yes. It simulates few-celled organisms and has challenges where the player is presented with an environment. You have to design an organism to survive in it, so that's pretty cool. Then we had Hannah Lore, who had a pick called Congressional Dish. This is a podcast, and it sounds to me like it is a politically oriented podcast, right? Yes. Last episode, The Warmonger's Plan. No one really knows what Donald Trump plans to do. So they talk about that. Okay. And then we have one from Johnny, our friend in Cambridge. Here, this is a good addressing. TWiVian Titans and polyparasitologists. Greetings of the season. The YouTube video might be of special interest to the good doctors Despommier and Griffin. Wonder of wonders, recycling, vertical farming, or easier, and all the raw material a household can grow for household and personal needs. Such wonder from mycelium. And the link is to a YouTube video. The fungi in your future. All the things you can make from fungi.
It's so cool. It's this leather substitute, and a technician in my lab does leather work. So I sent it to her, and she was just totally jazzed too. It's a very cool video.
So
really neat idea.
There's a San Francisco-based startup called MycoWorks, and he's making clothing and other things. That's cool.
Yeah, he seems like a fun guy.
Johnny Ballinger,
right.
Your Cambridge pediatrician and faithful listener, learner, and proud promoter. 9 degrees C, intermittent heavy rains, patchy fog and mist, breezy with winds from the east at 21 miles per hour. And our last is from Brendan, who writes, Dear Doctors TWiV, I am an ICU nurse practitioner. Not everyone in healthcare is a, quote, doctor, unquote. Yes, of course, you're absolutely right. With a passion for infectious disease, I have listened to every TWi derivative with glee and advocated for every healthcare professional. Actually, I encourage every non-healthcare professional, too, I know, to do so as well. As a side note, I have a 4-year-old daughter and a son on the way, and it encourages me to hear all of the contributors on your collection of podcasts. Hearing how many of them did not have a straight trajectory to their successful career encourages those of us who take the occasional left turn in their academic or personal life. I do appreciate hearing so many female voices in rigorous science so that I may point them out to my daughter as role models. Thank you for your time and devotion to your subject. I have a truly enjoyable pick of the week, the Long-Form Podcast, a podcast that features long-form writers and their personal stories. Featured TWiV favorite, science writer Carl Zimmer, episode 223, December 7th, 2016. Perhaps they would feature our beloved TWiV host in the future. Always nice when non-science-based media focus on science. Thank you, sirs and madams. Brendan. That's a cool name, the long form. We do long form here on TWiV.
Yes, we do. I noticed this is only a 1 hour. This is only a 1 hour podcast.
For us, it can't be too long.
That's a good tagline, Dickson. It can't be too long.
It can't be too long.
And that is TWiV 421. We are marching towards number 500, aren't we?
We are.
Wow.
You can find TWiV at microbe.tv and also on iTunes. But most people, I think, listen on their phones, in which case you get a podcast catcher and download them there. And we do like getting your questions and comments. We have almost caught up with our email. Amazing, isn't it?
Goodness.
We actually just had a few more on the queue today, and they're just a bunch more. Most of them are recommendations and so forth. So maybe next week we can finish up. And do think about supporting our work. You can go over to microbe.tv/contribute. And we have many ways. We have a Patreon thing there where you can subscribe for $1 a month. We have use the Amazon link and do your holiday shopping. It should be done by now, of course. If not, you go to the link, you click on it to get to Amazon, then all your purchases, we get a small percentage of that. It doesn't cost you anything more. And there are other ways as well. And what do we do with this money? Well, it will help us travel next year to record on location and get Dickson lunch because he doesn't eat otherwise.
You can tell by looking at him.
Yep.
We do.
Thank you, Alan.
Dickson Despommier is right here at Columbia, but you can also find him all over the web. It's amazing. Parasiteswithoutborders.com and thelivingriver.com are two examples of his lovely websites. Thank you, Dickson.
You're welcome, Vincent.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Alan Dove is at alandove.com. He's also on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
And our guest today, Nathan Letts, is from somewhere in North Carolina. Thank you for coming by, Nathan.
My pleasure. Thank you so much.
Did you have a good time?
I did.
Is it better in person than listening?
It's different.
It's different. It's an interesting flavor.
Yeah.
The acoustics are much better than listening as I commute to work.
That's for sure.
They are.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsor of this episode, CuriosityStream. And now, if you would like to win a brand new shrink-wrapped copy of Principles of Virology 4th edition, that's the latest edition. I was cleaning my office out today and I found an extra copy. I don't need it. If you're the 23rd emailer to say, I love viruses, it'll be yours.
And they love me.
And today is the 23rd of December. That's where that's coming from. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.